Hello, welcome to the spatial and multi-omics session. And it's my pleasure to introduce Professor Jing. He's from the Shanghai Institute of Biochemistry and Cell Biology. And he's from the last 10 years, he has been working on understanding molecular mechanisms of development, or focusing on mouse early embryonic development. And he has established many different technologies, including GeoSeq. And today he's going to tell us a story about lineage mechanisms. Professor Jing, please.
Okay, thank you for the introduction. And I also would like to thank Guoji and the organizing committee team inviting me to be here to present recent work on the early cell lineages and tissue organization in early mouse embryos. Oh. So we know the mouse development started from the fertilization and the zygote by embryonic day 3.5. So they form the blastocyst, have the first cell fate separation with the inner cell mass and the trophoblast. And by embryonic day 4.5, the inner cell mass further divided into the epiblast and the primitive endoderm. Then they implanted into the uterus and began the implantation development. By embryonic day 5.5, they form the egg cylinder shape and have the proximal and distal axis started. And by embryonic day 7.5, in the posterior region of the embryo, they form a structure called the pyramidal stick. So then have started the gastration. By 1 day later, at about 7.5 embryonic day, so they form 3 germ layers of ectoderm, mesoderm, and endoderm. Ectoderm later developed into nervous system and the skin area, surface ectoderm. And the mesoderm develop into the muscle, heart, blood, and others. And endoderm mainly in the internal organ like the lung, liver, pancreas, and intestine. So we can see in the in this 1 day gastration from embryonic day 6.5 to 7.5. So the epiblast from 660 cells proliferated into the 16,000 cells, increased the cell number of more than 20 times. And also they became the 3 germ layers. So this is a very rapid proliferation and cell fate determination. So this is why the UK development biologist Louis Wolpert said it is not the birth, not marriage or death, but gastration, which is truly the most important time in our life. So according to the classical view, the hypothesis said the epiblast through gastration in the anterior region, ectoderm became the ectoderm and then became the surface ectoderm and the neural ectoderm. In the posterior region, in the pyramidal stick, there is a mesoderm and endoderm. So they thought it's all from the epiblast. So we ask, what are the molecular controls underlying the lineage segregation of 3 germ layers in the post-implantation mouse embryos? So we try to set up a systematic and unbiased analysis of gastration transcriptome landscape. So we're cutting the day 7 misgastration mouse embryos. Then from the distal region to the proximal region, each is about 15 micrometers thick. And using a laser microdissection to anterior, posterior, left, and right for cutting about 20 cells. then do the RNA-seq and data analysis. So then that time we have quite some technical challenges on this method. So after about 2.5 years of struggle, we established this GeoSeq. So they can now, you can see now we can have this method established. And then this is an example of from a distal region to proximal region of the cutting. And then you can see we collected anterior and posterior, left and right, about 20 cells. So this is an example of collecting from single day 7 mouse embryos. We can collect about 42 samples. Each is about 20 cells. Then we have this one like 11 layers, and then anterior, posterior, left and right. So this looks like a cone, because in the most distal region, there's a small tissue, so we will collect the 2 samples. So this looks like a cone. We call it a cone blot. So this cone blot, if we do the validation with in situ hybridization, you can see this is a posterior expressed T gene. So digitalized like this one. This is a tRNA-seq, data. It looks like quite similar. So also we can reconstruct the 3D dimension, 3D models. So with this one, we found then this by embryonic day 7, the mouse embryo have 4 transcriptional domains: anterior, posterior, and the lateral, proximal, and distal region. Also, they have the signaling activities and also the cell fate determination. After this, we try to understand the time course of the gastrulation. So firstly with 6.5 to 7.5, but we know mouse embryo implanted in the embryonic day 4.5. So we collected 5 stages of embryonic day 5.5, 6.5, 6.5 to day 7, 7.5. And we collected internal ectoderm cells and outside of endoderm cells. Since the mesoderm only started by day 7, so we collected 2 stages of the mesoderm. This is what we collected in the real thing. So in the epiblast, we collect anterior and posterior regions. Endoderm also the anterior posterior region. But mesoderm, we collect by day 7, 1 region, and by day 7.5, we collect anterior mesoderm and posterior mesoderm. Each sample is about 5 to 40 cells. So then we can have this 3 germ layer cone blot. So you can see this is from distal region to a proximal region. And in the middle, this is epiblast or ectoderm layers. And this is here, yellow color is mesoderm, and outside is endoderm. So then we can see this T cell, T gene expression you can see in the proximal region, posterior of the primitive stick, and also MixL1, also from the posterior region. So, and other germ layers like SOX2 is ectoderm and anterior markers. And SOX17 is an endoderm marker. You can see it's mostly expressed in the outside of endoderm layers. So when we put this, finish all the samples collection, we put the embryonic day 5.5 to 7.5, all the samples together. We try to calculate the developmental trajectories, but we found the endoderm samples cannot merge with ectoderm and mesoderm cells. They're always separated. So then we try to collect this in 2015. In 2015, Austin Smith, the group published a paper with data of embryonic day 2.5, 3.5, 4.5, and 5.5 data. So we use their data with ours. So we merged from the embryonic day 2.5 to 7.5, 8 different stages. And also we use a pipeline of Regulon analysis. So Regulon is one TF and it's a downstream target together. So we are using 200 samples with more than 20,000 genes. We have more than 280 Regulons. With this Regulon activity, we do the further analysis. So then you can see we can see the 3 germ layers is well represented: ectoderm, mesoderm, endoderm, mesoderm, ectoderm, and also prometastatic pre-implantation and epiblast. So according to the old model, so people said endoderm and mesoderm have same progenitor, common progenitor, but we found the endoderm separated with mesoderm and ectoderm quite early. And also we can see this mesoderm and ectoderm have a common progenitor together separated. So there is all the Regulons can form 9 Regulon groups. So this 9 Regulon groups can separate 3 germ layers: mesoderm, ectoderm, and epiblast, prometastatic, endoderm, pre-implantation very well. So all this is, you can see very famous TFs like endoderm Regulon is a GATA cell TFs. And the inside, this is a downstream target regulated. So if we put all our different stages, 5 different stages, you can see they form the different, with the DEG expression, differential expression genes, they form the inside of different expression domains. By day embryonic day 7.5, they have 9 expression domains. So we use this DEG, and with Regulon activity, we can connect it all the samples together. So you can see they have a common progenitor of by day 3.5 from inner cell mass. But according to the old model, so the older 3 germ layers is coming from the early epiblast. But from our new model, you can see endoderm is separated quite early with mesoderm and endoderm from a primitive endoderm. And also mesoderm and ectoderm have a common progenitor here. So this is, so from this here, you can see the 9 Regulon groups have now derived for the early 3 germ layers. So mesoderm, endoderm separated quite early, and ectoderm and mesoderm have a common progenitor here. And also, of course, some mesoderm contribute to the endoderm also. So with this one, we can see from the old models, people propose the 3 germ layers, and ectoderm, mesoderm, endoderm are from the epiblast, and primitive endoderm mainly contribute to the extraembryonic endoderm. But from our new hypothesis, we propose the endoderm started separation with the primitive endoderm, then mainly contribute to the endoderm, and the mesoderm also contribute to endoderm, and ectoderm, mesoderm have a common progenitor. So also we have this set up a web portal for more than 20,000 genes. This more than 20,000 genes, if you can see, you can have the differential expression patterns for each gene, 2-dimension and 3-dimension also. So with this one, we after finish this, we ask whether we can establish a single cell spatial and transcriptome map for the early gastration, just like we can put whether the single cells into the GPS system. So we collaborated with John Maroni's lab. They published early last year of a gastration single cell. So we use their single cell just like the day 7.5 as an example. So we collected the single cells. Then we established the zip code gene with the different stages. So we have two more new stages. And then, so for this zip code genes, we can try to put the single cell into the different state position. So this is a cross-validation. We can see we collected the known lineage, known position of single cells, then put them back, do the single cell seq and put them back to suggest that this zip code mapping is quite okay. So then we can put the single cells into the different regions with their names. It's quite good. So it's about 90% of single cells can put back into the different regions. So like this mesoderm, you can see here is endoderm, right, in the region. And also we can put the single cells into the three-dimensional gastrulation. Here is a day 7.5 whole embryo single cell map. This is ectoderm, mesoderm, and endoderm. And also you can see through this one, we can see the different cell type, like the rostral neuroectoderm, they mainly expressed at the ectoderm, anterior ectoderm, and the nascent mesoderm in the posterior region of mesoderm and some part of the chromatic stick. So also we can see the gene expression with this one. So you can see the post-F1 genes is mainly in the ectoderm anterior region. The T genes in the posterior region of ectoderm and mesoderm. And also SOX17 is mainly in the endoderm outlayers. So we can have this single cell resolution of spatial and temporal map of mouse gastrulation for 5 stages. So through this one, we can see the gene expression like here, T cells with gradually they expanded and the expression in the posterior region. And posterior F1 gradually narrowed down into the anterior region of the day 7.5. So, but this work is not finished. So summary here is we established a method and then have the resources of more than 20,000 genes can be used by the communities. Also, we have this lineage separation trajectories and have started to build up a single cell spatial and temporal transcriptome map for the early embryos. So finally, I would like to thank the people working on it and our collaboration with Jing Donghan and John Maroni. Thank you for your attention. Hello?
A very, very nice talk. Anyone has a question, please type in the chat box. We have a question here. Could you expand on the algorithm or concept behind the spatial mapping of single cells?
We set up a new algorithm. So then this new algorithm can put back of the single cells into the 3-dimensional. Yes, we have a new algorithm, and this one is tested quite nice. Yes.
Oh, we have another question. He's asking, have you or are you considering doing this sequencing on human embryo too?
My lab, not yet. Yeah, probably others. Especially for the human, we are more interested in the gastrulation, but people knows because the ethical reasons, the human gastrulation embryo is not accessible. So we are not working on it yet.
Mhm. We have one more question. It's a very interesting one. Kathy is asking, can you also map the Regulon data spatially too?
Oh, this is a good question. We are not thinking about this yet. Now, this is a very good question. Yes, we can try, but not yet. Oh, yes.
Mhm. Last call for questions. Okay, we have 3 minutes. I'm going to ask a question then.
Yes.
Professor Jing, you know, you have two kind of data, right? One is from the this geosic spatial data, and now you're looking into the single cell data. Do you see any, you know, differences in terms of, you know, these two data sets?
Yes. I said, but I didn't have time to talk it here. We found with their annulation, like the published Nature in this single cell atlas. So they name it with different names. And we found about 90% of the cells can put onto the position with their names, with their annulation. But about 10% is not in the position with their names. So this one, we are now still working on it. Interestingly, I am thinking they probably have started early of the transcriptome before they move to the position.
Oh, okay. All right. Very interesting.